Morning, I'm Len Tesoriero. I'm a plant pathologist, and this video has been made as part of the Building Biosecurity and Resilience project within the New South Wales Vegetable Industry Project funded by the Australian Government and the New South Wales governments. Today, we're just going to look at recognising vegetable crop diseases, some very broad principles, and probably just give an example of one of the key diseases that you come across. One of the key issues associated with recognising diseases is getting that data, as opposed to the insects where you can usually see the insect itself and the damage. For the case of diseases, you're really just looking at symptoms initially. You can't really see the pathogens that are causing them because you really need microscopes of all sizes. So that field observation, looking at the patterns of the affected plants in the field is very important. And then having a clinical examination where you're looking for the key symptoms and which parts of the plants are affected. So that examination can include using a hand lens, which is usually about 10 times magnification, and then going back into a office or laboratory situation using a dissecting microscope where you might be at 40 to a little bit higher, but most of the time 40 times magnification for most microscopes. And then using a compound microscope, which generally is used in a more specialist environment, but can be used in an agronomy shop or something, getting up to about 100 to 200 times magnification, which is sufficient to be able to see a lot of the fungal structures that might be causing diseases. And then beyond that, it's really more specialised laboratory type work where you're culturing on agar media, using other forms of microscopy, such as an electron microscope to look at viruses, and serological things like dipsticks and lateral flow tests, very similar to what you might have been exposed to with the COVID situation. And those sorts of tests are available for a number of different plant viruses and a few bacteria. And then beyond that, it's really very, very specialised, amplifying DNA, sequencing DNA, and using other types of specialised equipment to identify plant pathogens. Back in the field, though, it's very important to distinguish between what might be a pathogen-related problem and things that might look a little bit like a disease, but on the plant. But when you look at the pattern in the field, you might be able to relate it back to other things. So, for example, on the top left there, you can see just the plants along one edge are affected. That's what that symbolises. And quite often that's a situation you might expect where, for example, herbicides being used along the edge of the crop or along the headland, and it's just drifted in and affected a few plants in from along each row. In the second one, the middle one at the top, you can see a more typical pattern for a disease where it may have started, for example, from a seedling infection that had been planted out, and it's then spread to a few other plants and so you get little patches of disease. You can also get that sometimes where an insect might have flown in and transmitted a virus, and then it's allowed for a bit of secondary spread around. So you'll get little patches of affected plants. Quite often they're fairly random throughout the crop. On the right-hand side at the top, you can see where complete rows are affected, and that's, again, less likely to be a pathogen-related thing and more likely to be something to do with, for example, where fertiliser hasn't been coming out of at transplanting, or even before where there might have been herbicide use. So it's affected a whole row or whole area of the crop along the rows. Down in the bottom left is probably another example of more likely to be a disease where there's been more secondary spread, so bigger patches, and that's more or less a development from that one at the middle at the top. The next one across, where sort of just a sector of the field is affected, sometimes from a corner, but can be anything from one area moving in. And that's probably more likely, again, to be a physiological thing associated, for example, where there's poorer drainage. So after rain events or flooding events, you can have an area of the field that doesn't drain so well, and all those plants will be affected in that area. And then finally, over on the bottom right is where you can see a sort of, a sector towards the edge of a crop where perhaps an insect has invaded, bringing in some disease or virus, for example. And then there's been secondary spread across the field, and it's sort of not a random distribution, but more or less something that seems to have moved from one end. Quite often, thrips infection or thrips causing virus infections with spotted wilt virus or one of the other tospoviruses can actually appear a bit like that. So it'll be more, I guess, where there's been a weed bank beside the crop, and the thrips have moved in from one edge, and then they've moved across and spread the virus along with them. So to see these sorts of things in the field, you really you can ground truth it, but really drones these days would be a much better way of getting that overview and understanding a bit more about that distribution in the field. Non-pathogen disorders are quite common in my diagnostic experience. Something about half of all the samples that arrive in a laboratory for diagnosis tend to be more likely to be non-pathogenic causes to problems. So in some cases, it's just to rule out that a pathogen might have caused some of those symptoms. And again, using your knowledge of varietal susceptibility and topography to target areas of the field is a good way of monitoring. So in some cases, in my experience, I've found that where there's more moisture sitting around in the morning, there may be some trees or something like that that are shading part of the paddock. Sometimes if there's a hill on the eastern side, there'll be more moisture or dew that sits around for a longer period of time, and that's where diseases will develop more easily than other areas of the crop. So you can target. You don't need to always be surveying the whole crop. And then knowing varieties too that are susceptible, and they can even be used strategically to monitor, so that if you know, for example, a particular variety of, in my experience when we were doing work in onions and downy mildew, we found some of the white skin varieties were much more susceptible and would show up downy mildew quicker than any of the other varieties. So it was always easier, particularly in large fields, to target the varieties that you know are more susceptible. And then one of the key things for diseases is there's so many of them. So you heard from Andy about the different pests and beneficials, and you're probably looking at, to say 20 or 30 different organisms there. But for diseases, there's many, many different diseases, and they come in all shapes and sizes. So the symptoms sometimes can look very similar. So it is important to use other information, and these pest guides are a useful way of at least seeing different images of diseases, which might be a bit confused if you're not aware of those particular things on different crops. So when we look at the types of plant pathogens causing diseases, we think about viruses, and as I said, they're very, very small, so they need very sophisticated microscopes of about 30,000 times magnification to see them. And tomato spotted wilt is one, but there's many different viruses, some spread by thrips like tomato spotted wilt and impatient necrotic spot virus, others that are spread by aphids. So a lot of the viruses that you might see on potatoes are spread by aphids. And then there's others that are spread by other things, sometimes transmitted just mechanically with humans, workers, machinery and tools. So that virus that's turned up in South Australia, tomato brown rugose fruit virus, for example, is highly contagious. it's closely related to viruses that have been around for a little while in Australia, like cucumber green mottle mosaic virus, which affects all the different cucurbit crops. And they're highly mechanically transmissible. In other words, they're contagious on workers' hands, tools, etc., and machinery. Bacteria, there's really only a few different families of bacteria, of which there are members that are plant pathogens. So most of the bacteria that are out there on leaves and in the soil and in the environment in general are fairly benign. Most of them don't cause diseases. But things like bacterial leaf spot diseases are caused by a few different bacteria. Sometimes they're quite specialised to different host plants or host plant families. So it is important when you're doing this sort of work in agronomy or horticulture that you're aware of the botany of the different crop species, and that helps in terms of understanding what plants may be affected by a particular pathogen, but also in terms of things like rotation crops and the like, as to whether or not they're going to be susceptible. So for these bacterial diseases, there's a few that cause things like galls, others that can cause vascular wilt diseases like bacterial canker in tomatoes, and which also affects capsicums a little bit. And things like bacterial wilt, which affect a wide range of plant species. In Australia, just generally, it's the solanaceous species that are affected. And then many different bacterial species or subspecies that are causing leaf spot diseases. Another group, and you may not have even heard of this group in that the word I've used is the plasmodiophorids. It's a group that I guess in the past we would have called them lower fungi, but they're not really fungal pathogens. They're actually more related to things like some of the algae and their little microorganisms. And they're a strange little group, but one of them is the pathogen, a Plasmodiophora, that causes clubroot in cabbages and related plants, and powdery scab, which is in potatoes. So quite an interesting group. They're things you might have heard of, slime molds, and they're in that group as well. So they're quite a strange group. But again, only a couple of them are considered to be plant pathogens of concern for us. Another group, and again, you may not have heard of this word, stramenopiles, or you might have heard of oomycetes, more commonly called water molds. So things like pythium and phytophthora. The downy mildews are in that group as well. They're actually related to those plasmodiophorids. And so again, in the old days, we would have called them lower fungi, but they're not really fungal pathogen, or they're not really fungi at all. And the other one that's in there is white blister or white rust. It's in that group as well. So pythium, phytophthora, downy mildews, and white blister are all in this group of oomycetes or stramenopiles. And then within the true fungi, there's three main groups. The zygomycetes, which you don't really need to worry about so much. There's only a couple that affect crops, and so far as I can think, most of them are not problems in vegetable crops, except one perhaps is a fungus that lives in the soil called pythium, and it actually is a vector for a couple of viruses that attack things like lettuces. So lettuce big vein is actually spread by one of these types of zygomycete fungi called pythium. Within the ascomycete group, that's a really big group, and there's lots and lots of different members of that group. And sclerotinia is one that I'm going to use as a bit of an example of a fungal pathogen that you're probably well aware of already, but we'll go through that in a little bit more detail later. And then the basidiomycetes, so they're the mushroom-type ones, which are, mostly non-pathogenic. They sometimes have strong associations with trees, and so you'll see those mushrooms appearing around the base of trees. But in the case of vegetable crops, really, Rhizoctonia probably is one of the main one, and the other one's sclerotium rot. So sclerotium rolsii, you might know, which causes a stem rot. And Rhizoctonia obviously causes damping off and lots of different diseases, even fruit rots, and gets on a wide range of things. And again, there's many different Rhizoctonias, as there are many different basidiomycetes. So we're looking really at, when we sort of add just the fungi together, it's probably over 100 different plant pathogens in that whole group. And then finally, the nematodes, or roundworms, or eelworms, as they used to be called. And probably the most common one we encounter is root knot nematodes. And of those, there are several species, and you might have heard of guava root knot nematode that has come into Australia in more recent years. But there's about 4 different species of those. And then within each species, there are different subspecies, some for which there are plant varieties with resistance. So there's, again, quite a complex interaction between the actual form of the nematode and the actual variety of plant that you can use. So again, it's not a simple one, but the symptoms of root knot at least are pretty clear. And as I said, the diagnostics is assisted by your knowledge of botany. So, for example, if you're looking at a carrot and you know that it's a member of the Apiaceae, and you have other members of the Apiaceae that could be affected by the same or very similar pathogens, so you might find in parsley and coriander and a few other things like that parsnips might be susceptible to similar diseases. And similarly for, say, the solanaceous family, as I mentioned earlier, they tend to be things like spotted wilt virus might affect quite a range of different solanaceous hosts. I like to put this slide up because it shows you how diseases are really just one aspect of things that affect plant health. And quite often plant pathologists will put up what we call the disease triangle, where we just talk about the actual plant, the disease or the pathogen, and the environment. But when we look at the environment, it's made up of many different components. So you can think of the environment as just the weather conditions, the overall climate in which you're growing. But it is important to think about the soil condition itself, and that includes aspects to the soil condition, such as the biology in the soil, as well as the physical structure and its chemical structure. With regards to weeds, we know that a lot of weeds can host plant diseases, sometimes viruses, but also some of the fungal pathogens or even the bacterial pathogens. We can think of, again, the pests that I mentioned that are vectoring diseases, and in some cases just causing damage, which allows some of the opportunistic plant pathogens to enter. So think about things like some of the soft rot bacteria that can be secondary infections after there's been some insect chewing injury or the like, and the bacterial infection just follows from that. Plant nutrition can be vitally important in terms of how it affects plant health on its own, but it also interacts with diseases. And one of the, I guess, strongest examples I know of is fusarium wilt in certain crops, and I've seen it certainly in solanaceous vegetables, but also in the cucurbits, in cucumbers and melons, where using excess ammonium form of nitrogen can actually increase the susceptibility and the symptom development of fusarium wilt. Just on that one, it's ironic that there's another vascular wilt disease called verticillium wilt, which has actually affected the opposite way. So that ammonium nitrogen actually suppresses verticillium wilt. So in one case, you've got it increasing the susceptibility and the symptom development, and in the other case, for verticillium at least, it suppresses the disease development. And of course, your use to pesticides is important. Obviously, using the wrong rate of pesticides can either lead to escape of the pathogen if it's too low, or using a high rate, a rate that's above the label rate can sometimes cause phytotoxicity, and then that damage can allow some of the pathogens to enter. And then that bottom there on the left-hand side, plant genetic resistance, is obviously really important in choosing a variety that's possibly got resistance or not as susceptible to some of the plant pathogens. And again, work with the seed industry to deal with that one. Okay, so let's look at an example of a fungal pathogen, and one that you're probably very familiar with, Sclerotinia rot, and it's caused by the fungal pathogens Sclerotinia sclerotiorum and Sclerotinia minor. And they're very easy to distinguish just from the size of those black resting structures, the sclerotia, which are the survival structures in the soil. The larger ones being Sclerotinia sclerotiorum, and they form those globular, irregular lobe structures that can look a bit like a rat feces. And Sclerotinia minor, that smaller sclerotia, which are roughly spherical and about the size of a match head. So here's an example of Sclerotinia rot on different plant species. You can see a lettuce there up on the top left, and those black structures with the white woolly growth. That cottony fungal growth is very typical of Sclerotinia. Tomato there, where you can see a stem infection with the black sclerotia forming. The cucumber on the top right, a fruit infection, and also just below it, a stem infection. Excuse me. And you can see at the bottom there's a tomato which is infected, and over on the right-hand side, a cherry infected. So a wide host range, over 400 hosts. Over on the bottom left, you can see that mushroom-like structure, and I'll talk a little bit about that in the next couple of slides. Here's an image of a badly affected lettuce crop where the plants are dropping, so that disease is very commonly seen in lettuce. And again, because of the fact that those sclerotia can survive for long periods of time and it's got such a wide host range, you can end up with individual fields with high levels of disease such as this. So this lettuce drop, and in this case it's Sclerotinia minor, the lettuce itself in this case is just melted away, and you can see that there's hardly a trace of it left there, but those black sclerotia sitting on the surface of the soil. So let's look at the life cycle. Where should we begin? Well, we begin with the sclerotia. So let's start with the top right-hand side where you've got a lettuce there, those black sclerotia. As I said, quite often the actual plant tissue will just dissolve away and leave the sclerotia on the soil surface. Now, from that, you can either get infection of the new crop or the neighbouring plants via the germinating sclerotia in the soil, which can further cause disease. So it just produces that woolly hyphae, that white woolly hyphae, which grows across the surface and will attack the neighbouring plant through the base of the stem or through the root system. Or alternatively, those mushroom-like structures, so in this case, these little disc-shaped things have got little pores in them in which sexual reproduction occurs, and spores are produced which are released forcefully into the air, which can then float in air currents and attack aerial parts of the plant. So you can get infections, for example, on just above it there in that cucumber on the fruit, or through the flower and then into the fruit. And similarly, in the blossoms of something like cherries or onto the cherry fruit itself. So the this aspect to sclerotinia infection allows it to spread further than it would if it was just moving from plant to plant via the soil or on the soil surface itself. And here's a pretty bad example of a cabbage crop infected with, and in this case, abandoned due to sclerotinia infection. And it's probably worth noting that even if prices are low and you don't want to harvest all of your crop, by leaving the affected plants there, all you're doing is growing the fungus that's going to survive in the soil for the next crop. So it is important to try and remove that crop as quickly as you can. Sclerotinia can also cause a post-harvest problem called nesting, and this is an example, and you can again see the furry growth in this fancy lettuce crop here, and you can see those little white structures at the base of the plant, which is the formation, the early formation of those black sclerotia. So in terms of interventions for sclerotinia, and this is probably true for a lot of different fungal pathogens, but some of them specific here for the sclerotinia fungus. In terms of preventing disease development, it's trying to stop the production of those sclerotia if you can, or at least allowing those sclerotia to be parasitized. And there are some beneficial fungi such as trichodermas and a thing called Coniotherium, which is not unfortunately available commercially in Australia. But the trichodermas which actually can parasitize those sclerotia. So that's something that can be used as a way of helping to control the carryover of those sclerotial bodies but from crop to crop. And I know that in some of our experimental work, and it's something we'll probably do a bit more of, we've been looking at, I guess, the best timing for the intervention with trichoderma. And we found that it's probably best to put it on the crop residue before you turn it in, because you're really wanting to get the trichoderma to attack the sclerotia. And so if you did it just before you turn the crop residue in, you've got a better chance that the trichoderma is going to find its way into those sclerotia and colonise it and kill it. Some of the streptomycete bacteria are also potential in that area and probably requires a bit more research. By and large, growers have pretty much relied on the timing of fungicides, and usually for most cropping things like lettuce and cabbages and the like, it's important to get that fungicide spray on just after transplanting or after emergence, so that you've got that early intervention. Most chemicals, and for that matter biologicals, are really much better applied as preventatives than a curative. So it's important as an insurance policy, as much as anything, to get that chemical spray on early so that you're preventing any disease build-up. The key interventions include, as I said, the removal of plant debris at the end of the crop. Most of those sclerotia are in the top 8 centimetres of soil. They actually respire, so that they will actually die if they're deep buried. So if you bury them deeper, they'll actually die. They won't actually survive as well. One silver lining of flooding is the fact that it deprives the sclerotia of oxygen. So it's not something that's always desirable, although it's kind of interesting that when I read stuff from the United States, they use a strategy where they do flood the soil. And I'm not recommending it, because basically flooding the soil deprives the soil of oxygen, which kills off a lot of beneficial organisms in the soil as well. So it's not something that I think is desirable, but it might be one silver lining to actually, if you're in a flooded area or have had a flooded come through. It may bring sclerotia in with the floodwater, but it might also kill a lot of the sclerotia that are in the soil. As I said, Deploughing might be one way of asphyxiating the sclerotia, and using rotations and with things like maize and cereals and alliums, beet, spinach, some cucurbits, like I showed you, cucumbers are affected, but generally it's not a major problem in cucurbit crops, but certainly things like a cereal or maize rotation is probably the best thing you can do. So I might wind it up there. As I said, there's many different diseases. It probably would take a whole raft of these sorts of presentations to go through each one in detail. So I thought if we just chose one today, that'll give you a little bit of a taste for how to look at the life cycle and the interventions and where and how to go about it. Thanks for your time.